If you're wondering why this took so long to get up, you guys, it's because after I made the first video that was an hour and a half long, I deleted it on accident. Budge. So let's do this again. Another hour and a half of review. It's great. Basically what I was going over last time was saying, we have chapters 19, 25, 26, and 27. Scratching? No, I'm not. So then basically 25 stations divided by 4, it'd be 6, 6, 6, and 7 for each chapter. How many stations per chapter? And then since we do not have the lecture part after, or the lab after the practical, Pesci is going to make the practical just a little bit longer this time, and then so you're going to get your normal 25 stations, but plus the genetics problems that I told you in class that she was going to give me for this practical. She's going to make her own set, and then she's going to give it to me to give it to you guys. Alright, so let me start all the way over again. Goodness, this sucks. Okay. Alright, I'm on page 504. I want you guys to look in exercise 19.1 and highlight the word hypothalamus. I would like for you to know what effect it has on the pituitary gland. Then on page 505, you see that that big table? You are going to highlight the column, everything under the column labeled pituitary gland. You're going to highlight everything under the column labeled cells. Highlight everything under the column labeled description. Highlight everything under the column labeled hormones produced. And everything under the column labeled action of pituitary hormone. Then on page 506, you are going to highlight figure 19.2, as we did view that on the microscope. So no chromophobe, basophil, and acidophils. Be able to differentiate between them on the microscope. Then on number 6, highlight the word pituocytes. Know that they are derived from glial cells. And know what hormones they produce. So oxytocin, antidiuretic, ADH, vasopressin. Then on page 507, highlight melatonin. Know what melatonin is. Know its effects on humans and on other organisms. And then also circle picture figure 19.4. If I say to view a slide or like highlight a slide that you guys didn't view in class because we didn't have the slide, then I would still know it anyway, because if I can't find that slide, then I'm just going to wind up pulling one of these from like Google or something, and I'm going to pull this exact image. It's going to look exactly like that, or I'll probably just go scan, copy this, take a picture of it. I don't know. But it's going to look like this. Like, if you study these concepts, you'll get it. So, circle figure 19.4, highlight pineal gland and pinealocytes. Be able to identify those in that slide. Then on page 508, highlight the slide labeled B, thyroid gland, because we did go over that. I would like for you to know follicular cell, parafollicular cell, thyroid follicle, and the colloid within the thyroid follicle. So highlight all of those. Now you're probably wondering, well, how are we going to know the difference between follicular and parafollicular? Well, follicular is it's the direct outside lining of the thyroid follicle. So it's the ones that circle the thyroid follicle. Now, parafollicular is often like interstitial space somewhere. It's not in an interstitial space somewhere. It's not going to be in that direct lining of the thyroid follicle. So if I'm pointing to a cell that's surrounded by a bunch of little, like, also dot cells, then it's parafollicular. If it's the cell inside, like, the lining outside of the thyroid follicle, then it's follicular. So I would highlight that slide. Then, on page 509, the whole entire table, except for the last 2 columns. Highlight it. Know it. Cell types, description, hormones produced, and action of hormone. Then on exercise 19.4, the adrenal gland, catecholamine hormones, highlight that, know what that is in that section right there. Then on page 510, sorry if I'm going fast, you guys can just pause this. I did just do this, so I just kind of want to get through it. But if I miss anything, I mean, if I'm going too fast, you know, you can rewind and pause and all that good. All right, so then on page 510, I would study, it's table 19.3. I would study, so know all of the zones in the adrenal cortex, glomerulosa, fasciculata, reticularis. Then know the cells and the medulla, so the chromaffin cells. Then I want you to know all of their hormones produced. I don't care what type it is. I just want to know if, like, for zona glomerulosa, all you have to say is aldosterone. For fasciculata, all you have to say is cortisol. For reticularis, all you have to say is androgens. And then norepinephrine and epinephrine for chromaffin. Then I want you to highlight the whole column that says action of hormones. So I don't care about the description and I don't care about the origin in that table. Then, on page 512, oh, this one's good. So it's table 19.4, the histology of the pancreatic isolates. So I want you to highlight the column labeled cell types, know all of them. I want you to know the descriptions, but only in like reference to how much it makes up of isolate cells. So like 30% for alpha, 65 for beta, 4 for delta, and then roughly like 1 for F, which are rare. Know that. Then the hormones produced by all of those. And I said also action of hormone. And that's pretty much it. And then we're going on. Okay, so on page 514, you highlight the organs, because we did this on the cat. So highlight the thyroid and the parathyroid, not the parathyroid, we can't see the parathyroid, right? So just highlight the thyroid, highlight the thymus, highlight the testes and the ovaries, because remember I did find those 2 cats and I labeled them. I found a good girl and a good guy, and the girl had like really nice big ovaries and the guy was good. And then I would also obviously just label pancreas. I could ask this on the cat. Then on page 515, it's a picture of the brain. I could use like a half section of one of those plastic brain models we have. This might be like a kind of easier station if I just want to throw one in. But I would highlight 12, 13, 14, and 15. So hypothalamus, pineal gland, pituitary, anterior pituitary, and posterior pituitary. So I would know those. Then metabolism, we're just going to pass that because I think I already have like more than enough for... Yeah, I have a lot. And then we're going on to chapter 25. So that was the urinary system. Okay. Basically in this one, okay, on page 688, highlight everything under, well, in that table 25.1, highlight everything under the column labeled structure. All of it. Then, for epitheliums, I only want you to know the epithelium of the visceral layer of the glomerular capsule and the parietal layer of the glomerular capsule. Then, I want you to know the epitheliums of the thick and thin segments of the descending limb and the thick and thin segments of the ascending limb. When it comes to parietal and visceral, know which one's inner, which one's outer. PCT, it's site of most reabsorption and secretion. Know that. DCT is involved in reabsorption and secretion. Why am I stuttering? Then on page 689, I want you to go over that whole model labeled B, because we do have that, so I can definitely use that. So distal convoluted tubule, proximal convoluted tubule, nephron loop, renal corpuscle, cortico-medullary junction. Well, what's wrong with me? Of the nephron loop, no, the ascending and descending limb, arcuate veins, so the artery and the vessel, collecting tubules, collecting ducts, papillary duct. Remember, we couldn't find renal papilla on the model, so you're good on that. You don't need to know it on the model. Glomerular capsule, which is just pretty much the same thing as a renal corpuscle, but anyway. be able to identify that on the model. On page 690 and 691, it's 2 slides of the renal cortex. Remember I said how to know the difference between the cortex and the medulla. That's really important, because I could do that to you guys again. I can give you a slide and ask you, is this the cortex or is this the medulla? And you have to be able to differentiate between the two. So to do that, I'm not going to give you the answer, but to do that, look at the slide on 690 and then look at the slide on 692 and tell me what you see and what you don't see. Or you can just go to the table and see what's in each section because it says the medulla and the cortex. So for that figure label 25.2, highlight capsular space, glomerular capillaries, afferent arterial. Afferent arterial always attaches to the glomerulus, always. I'm not just going to be talking about a duct somewhere that's not attached to a glomerulus. Then also on the figure 25.3, renal corpuscle, highlight DCT, PCT. The difference between those two, or how to tell the difference, is that they both have microvilli, but like DCT, you can't see it at all. It's really only kind of like on the inner part. It's just not as dense. It looks more smooth, as you can see in that picture. And then on PCT, you can see the lines, like, right, all through it. It's very dense. That's how you would be able to tell the difference. But, like, don't get it wrong. Like, they do both have microvilli. It's just one has a lot more than the other. So that's how you would tell the difference between a PCT and a DCT, if I pointed to one. And also, like I said, know that fun fact about microvilli. Like, they both have it. Know that. Page 692, highlight the, so now we're talking about the medulla, right? Yeah, the medulla. Highlight the B slide, so the last one. And then highlight thin limbs of nephron loop, thick limbs of nephron loop. That's easy to remember because it's the circular layers with cells in them. And they're the only circular layers with cells in them. Yes. So the ones with cells in them that are super, super thin are obviously the thin. And then the ones with cells in them that are super, super thick, like simple cuboidal, are obviously the thick ones. Then the ones that have a circular lining but no cells in them are vasa rectas. So know that. Collecting ducts, be able to tell the difference between that. A collecting duct, seeing the difference between that and then the nephron loops, thick or thin, well, no, just thick, is that the collecting ducts, it is extremely defined, simple cuboidal epithelium. And I know I just said simple cuboidal epithelium for the thick, but it's not. It looks like it. It's just like, it's not as structured. It's not like, here's a cube, here's a cell, here's a cube, here's a cell, here's a cube. It's like, oh, here's a cube, here's a cell. Here's a cube, here's a cell. Here's a cube, here's a cell. You know what I'm saying? Like, it's not as structured as a collecting duct. As you can see in that picture, that actually helps you a lot. So highlight that. Then on page 693, I would circle the image labeled A. Even if we didn't get that, if you didn't see it, I did use it on the quiz. I can definitely take it from Google. I want you to know the inner longitudinal muscle, outer circular muscle, adventita. Know what it is. It's the ureters. Then we also did the kidney dissection. I know there wasn't a lot to see, but what we could see, study that. So it was renal sinuses. We were able to see the cortex, the medulla, the renal lobe. I can ask that, and then the ureter. If there was anything else, remember to study it because if I can see it, then I might ask it. Then on the same page, that's 695. It's exercise 25.6. Label that first line. It says the kidneys receive approximately 20 to 25% of blood pumped cardiac output by the heart each minute. Also, on the second part of that exercise, it says the glomerulus is the first capillary bed, which is the site of filtration. Know that. On page 696, then it says the efferent arterial then leads to the second capillary bed, which is the paratubular capillaries or the vasa recta. Know that, and that they're the site of exchange of fluids. electrolytes, gases, all that good stuff. Then I would definitely circle figure 25.8, just circle the whole thing, because it shows how the blood flows specifically. So how it goes through the kidneys. It's going to go through the renal artery, then all the other little arteries, right? Then it's going to go through the afferent arteriole to get to the glomerulus, where it's got to do its exchange in the capillaries and all that stuff. Then it's going to exit through the efferent, and then go through all the veins, out through the renal vein, and then it's out of the kidney. So that's blood flow. But then I also want you to know, like, the process of urine, because urine doesn't go through the veins. So how does it start? So it starts when the exchange is going on in, when the exchange is going on in the capillary beds. It leaks over. Because it goes in through afferent arterioles, and then it stays in here, and then it goes out. But when it's in here, it leaks over into the Bowman space, and then when it's in that space, it connects to the PCT. Well, then it goes through PCT, and it goes down the nephron loop, up DCT, to a collecting tubule, into the collecting duct, because that's where it drains, down the collecting duct to the let me make sure. Yeah, papillary duct, and then out through the renal papillae. That model, like I said, doesn't have the renal papillae, so don't worry about that, but do know it. So we want to know, like, that whole flow of blood, also the urine formation. Then, so on page 697, I want you to circle that all of A. I want you to know everything about that, to be able to label a kidney like that. A dissected version. Also of B. I don't have that exact model, but the structures are also the same. If you study that, you'll still understand it on another model. Then on 699, just be able to identify, because we did identify the ureters and the urinary bladder in the cat, be able to find those in the cat again, or know what I'm talking about, know the difference between a ureter and what I'm talking about, like an ovary, if I have the girl cat or not. Also, so we're on 699. Now exercise 25.9. Know the internal urethral sphincter and the external. Which control is which, voluntary, involuntary. Know that the trigone is the area of the floor of the urinary bladder, and why is it clinically important? It'll tell you there. Then on 700, know the male urethra has 3 parts. It's in that part 4 right there. So prostatic, membranous, spongy. Then 701, highlight it all. It's the male and the female's mid-sagittal, genital area, section, urinary, all that good stuff. Yeah, but I would know all of that. It's just about, it actually just has to do with the urinary stuff. So just know all of that. And then for urine formation on page 702, basically all of this is going to sum it up and tell you what I just told you. So what happens. So you can read all of this so you can get a better understanding if you don't already kind of understand it. But that's basically, I don't care about all these vocabs in here. I'm not going to test you over this vocab part, only what I just could say, basically. But do know the 3 parts. So filtration, which is in this first part of the, so this first part, it's up here at the top, and then reabsorption is in the second part. It's at the bottom of that paragraph, and then on the last page of 703, it says secretion. So just know those. Also, did I go over edema? I did. It's in the concept connection. So just highlight, there's a part, it's the second to last paragraph. It says, damage to the filtration membrane may allow larger molecules, including albumin, to enter the filtrate. Because there are no mechanisms for reabsorbing albumin within the nephron, any albumin that is filtered ends up secreted in, or excreted in the urine. Then highlight the last sentence of that whole concept connection. So the result in fluid accumulation in the interstitial space surrounding systemic capillary beds is called edema, like when body parts swell up. Then for the p-test, remember the p-test, on page 705, it tells you what normal results are. So like for specific gravity, it tells you the normal results, pH, if there's signs, traces of glucose, negative ketones, whatever, what's normal, what's not, study that. I can always take one of those unknowns and take a strip and do it myself and ask you, well, what's the patient's problem in this situation? So I would like you to memorize that. Sorry, I just got something in my eye. Also know, what does the presence of white blood cells indicate? And know what each of these are. Like, what is specific gravity? I mean, obviously, we know the number, but what does it mean if you have a high number or a low number? Like, what's going on with your body? Are you... Tell me, you know? I said this in class. But pretty much just specific gravity, because we kind of know what the other ones mean. Well, yeah, glucose, obviously. Protein, obviously. What is protein? Okay, so that's it for that one. Then we're going to move on to chapter 26, or the part of 26 that we didn't do last time. In this case, I guess I didn't go over the... Anyway, we're on 741, and this is... Okay, so basically this is the one we didn't get enough time to get to. So I told you guys just to read over it on your own time, and then just forget about the experiments. So in this case, I would say, since it's really only like 2 pages, study the whole thing. Basically, just study that whole, it's page 741 to 743. Let's just say that. And basically, I would like you to know everything on the first page. So everything under digestive physiology, everything in the clinical view, everything in table 26.10. Then on 742, I would know everything on the top of the table. Everything on top of the table. So even the concept connection. The table, I'll leave it out. I'm being nice. And then on 743, know the clinical view and the concept connection. And then stop before you get to exercise 26.14, because we don't need to do the exercises. And that's pretty much it for that chapter. This is probably only going to be like 3 stations, actually. So I lied. It's not 6, 6, 6, 7. It's going to be a little bit more for the big chapters, and then probably 3 for this. So, so like 7, 7, 3, 8. There we go. Okay, and then 27. So now we're in the last stage. Oh my gosh, I did this in 23 minutes. I don't know how I did that. Thank you. All right, so on page 754, basically highlight everything in the column labeled cell name, oocyte stage. I want you to know the description and functions of each oocyte for sure. I want you to know the stage of mitosis they're in each phase, and then I want you to know the ploidy. So when do they switch to a haploid? When are they diploid, and when do they switch to haploid? Then on page 755, I really love this picture of the ovary. Since we don't have a model of the ovary, I'm just going to take this from Google. But I would probably definitely use it, so keep your eyes out, study it. Basically highlight premortal follicle, primary follicle, and then in the primary follicle, it's also labeled primary oocyte. Highlight that. Highlight secondary follicle, the zona pellucida, and the antrum. Then highlight vesicular follicle, antrum, secondary oocyte, zona pellucida. Then you see, obviously, the egg is rupturing from the follicle in that picture. So know the corona radiata, zona pellucida, and the ovulated secondary oocyte. Then I'm not going to ask you developing corpus luteum. That's fine. But corpus luteum, know that, and then know, obviously, when fertilization and implantation do not occur, it's going to turn into scar tissue, which is then called the corpus albicans. What hormones drop? Think about that too. Okay, then on page 756, so basically, I know we didn't have pictures. I didn't have slide pictures of all of these, but I could take these from Google too. Like I said, if I do, it'll be the exact pictures. But it'll be like so similar. So I would, in table 27.2, I would know all of those slides. Be able to identify that, because I want to be able to give you a slide and say, what stage is this follicle in? And I want you to tell me which one it's in just by looking at it. Now, to tell the difference between a secondary and a vesicular, I told you guys in class, they both have antrums. Antrums get created in the secondary, but the antrum is so skinny and very C-shaped in the secondary follicle. But then when it goes to the vesicular, it fills up and it swells. And it's still pretty curved, but it's not as like C-shaped and defined. So I would be able to recognize what follicle stage the slide's going to be in. Then on figure 27.3, I would circle that whole thing. I would highlight granulosa cells, antrum, corona radiata, zona pellucida, and then secondary oocyte and cumulus oophorus. Because I do think we had a vesicular follicle. Even if we didn't, Google. So know that. Then on page 758, highlight slide B, mucosal folds, highlight secretory cells, highlight, and simple columnar ciliated epithelium. Highlights. On page 760, I want you to know table 27.5. Just know it. Everything about it. It's the 3 phases of the menstrual cycle. You've got menstruation, proliferation, and you've got secretory. Know the days and know what's going on. Like, I want you to know everything about that table pretty much. Then also table 26.7. So in this table, know pretty much all of the wall layers, because these are the layers of the uterus. So know all of that column, and then description and function. Then, I mean, I would, this could be an easy one. So on 671, there's that slide. I mean, you can highlight basically epithelium, lamina propria, and muscularis. And that's the vaginal wall. So know that that's the vaginal wall. Or I might just, I don't know if I'm going to cover slides. I don't know if I'm covering slides yet. I'll figure it out. For that one, I probably won't. Even if I do cover, like, for that one, I won't probably, because that's hard. So yeah, I guess just know those layers. Then, for the male, ooh, that's a good one. Highlight B. This is on page 762. And if you did see C, which I don't think you did, but if you did, highlight it. If you didn't see it, I'm not going to use Google on that one, so you're good. But then in table 27.7, I want you to highlight the column labeled cell name. Note all of those. These are the developmental stages of sperm. It's very important. I want you to know the description and function. Well, not really. Just the stage of mitosis meiosis. But like, I kind of want you to know. No, just that, and then also the ploidy on this one. So everything under those columns. Know the difference between the accessory cells of the testes, so the interstitial cells and the I just call them Sertoli cells, but they're sustentacular. Then, and that was on page 763, so just know the difference between those two. Then, I would know the on page 764, table 27.9, highlight the column labeled structure and then highlight the column labeled function. So know the structures, know the functions. Then on the histology of the ductus deferens, know those slides. Just be able to identify them, and yeah, pretty much everything inside of it's simple and easy. Lumen, mucosa, yeah. So be able to identify those. Know that it's the ductus deferens. That will be covered. So you need to know the difference between that and the ureters as well. Seminal vesicles, I like that too, but that's also easy. All you can really do is those three, so just be able to identify that that's the seminal vesicles. That will be covered. If there was a prostate gland, definitely study we'll actually just study the prostate gland in general, because that could be Google. Know the prostatic caliculi, and then the tubulo-alveolar glands. And I don't think I think I will have that one hidden, because it is very apparent what's going on there. Basically, prostatic caliculi is what happens over time, like as a man gets older in his prostate. So those things shouldn't even be there. I mean, that's very obvious what that is. So I would keep that covered. Histology of the penis. We did have this one. Yes, study this one. Know the dorsal vein, urethra, the corpus cavernosum, superficial fascia, corpus spongiosum. Yeah, that's it. Then I don't really care about the ligaments too much. When it comes to page 770, I would like for you guys to know fimbria, body of uterus, or not, broad ligaments, vagina, endometrium, myometrium, parametrium, what's internal OS? Internal OS, cervical canal, and external OS. The isthma, ampulla, and infundibulum of the uterine tubes. The fundus of uterus. That, just study it like as if it was like on a model like that. Because that's another, if I don't find the model for that, which I don't think I will, that's another Google. Then on page 771, everything. Just study it all, because I don't honestly know. Like, let me think. Yeah, I would just study it all. It's a female, yeah. And then also on, well, hold on, we'll go up there. I would also study 773, all of that, because I can use a picture of that if I can't find a model of the breast. Then on page 775, I really like this picture. I think I could ask you to draw it out. I would want to see basically let's see. Ureters, that's cool. Urinary bladder. Just know these. Know them. Be able to identify them. Be able to draw it if you could. Whatever 3 was, because I don't have it highlighted. I don't want to say something wrong. I'm pretty sure I know what it is. Then also the ejaculatory duct. So the ductus deferens and number 3 are both secreting, and they're going to go through the ejaculatory duct, right? So highlight ejaculatory duct. Then that's going to transfer into the prostatic urethra, but no prostate gland and prostatic urethra. Then bulbourethral glands. Then I don't care about that. Whatever 9 was, I think that's the urethra. I don't know I don't have it written. Sorry. 10 ductus deferens. We know that. Yeah. So no ductus deferens. Epididymis, testes. So it's testes, epididymis, vas deferens, ejaculatory ducts, prostatic urethra, and then through the urethra. I would also know bulb of penis, crus of penis, I guess. I like that thing. Yeah. Corpus spongiosum, glans penis, and spongy urethra. Oh, basically everything. Except for like 2 and 8. Yeah. Okay. Then on 776. Anus, corpus cavernosum, corpus spongiosum, ductus deferens, ampulla and ductus deferens, ejaculatory duct, epididymis, glans penis, internal urethral sphincter, membranous urethra, penis. Don't really think I care for the prepuce, or I do care about prostate gland, prostatic urethra, pubis symphysis, rectum, scrotum, seminal vesicle. Seminiferous tubules. Where is that one? Okay, yeah, seminiferous tubules. No, I don't care about that. Spongy urethra, I guess. Testes. Urinary bladder. Ureter. That's it. 777. Know what gametogenesis is, so highlight that word that's bolded. Know all the phases of meiosis. That's easy. You guys have already pretty much, you've already studied this. Know the amount of human somatic cells contain how many pairs of chromosomes. How many are, that's it. And then how many are autosomes and how many are sex chromosomes. I would study the word bolded fertilization, everything after that. And then also when it says when a sperm has fertilized an oocyte, the resulting cell is called a zygote, is once again a diploid. 2 haploids make a diploid. Know that. Oogenesis on page 778. Know what that is. Then on 779, know this whole, when do these cells go through mitosis? It's before birth. It's in the embryonic period. So know that. Then know that the primary oocyte basically stops in prophase 1. And it doesn't start back up again until puberty. And then when it starts back in puberty, then know that meiosis 2 stops until fertilization happens. Yeah. So basically, study that chart on 779. Know it. Then on 780, I would like for you to know all of these 4. So the first of these, whatever these are, these ones. So this first one is the ovarian cycle. The next one is like the amount of FSH and LH that's being like secreted and at what times. And then you've got progesterone, estrogen, and the uterine cycle. So basically all of these line up. They're all 1 through 28, as you can see. So be able to know, oh, when is LH spiked? Like what, in comparison to the ovarian cycle, what phase is it? Like what phase is it going in? You know, just know, like I said, all these charts. For the ovarian cycle specifically, though, definitely know what's going on with the follicles in the follicular phase, ovulation, and the luteal phase. Know the difference. Then on page 781, spermatogenesis, highlight that. Spermiogenesis. And I think that's it. So in terms of genetics problems, I'm not too sure exactly what she's going to do or what she's going to put. I would study these things to help you guys out. So I would study this first one in terms of knowing like vocab and all that, just the proper words. I mean, you guys said you already know this, and I believe you, but like if you just need to like freshen up on some vocab words, I don't know what she's gonna ask, but I would just go over the vocab for sure. Then I know you guys gave this to me, but I know some of you guys did pick up 2 copies. So if you do have another copy, just go over it. If you don't have this, it's totally fine. You can still just go over this thing that you guys definitely should have. This is all your questions that are actually due on Tuesday anyway. So if you go over all this, you really should be good. Even if, I don't know if you guys did it or if you didn't, not on me. But I do think that these are going to prepare you for the questions she's going to get, because I'm pretty sure they're Punnett squares. Yeah. So I would say study those 3 things. As in terms of what's due, I told you guys that I changed the extra credit up. So extra credit for the final was either going to be this packet and this packet, or, and one's 1 and one's 1. So you can actually just turn in 1 for 1 point if you want to do that. Turn in both for 2. You can't turn in these 2 and this one for 4 points. That's not going to happen. So you could do that, or you could just get 2 points for filling out this packet. Principles to heredity. So it's up to you guys, whatever you want to do. They're both pretty freaking thick. These ones look actually the thickest, but if you just want to like basically do one of these and get 1 point, you can. But like I said, you can't do all of these and get 4. She would kill me. Then what else? The pig. I would also study the pig. We got to it in the second lab. So second lab, if you haven't already sent that, please send that. Thank you guys. For the pig, basically all I want you to know is how to, based off of measurements, which is on this first page of the pig thingy. I told you all to pick this up. I hope you did. So on the first page, there's this little part right here. Like right here, this part. Highlight all of that. it basically says how big the, how many days old the pig is compared up to the size of it. So like highlight that section as you see I did. It says 21 days, the pig should be 11 millimeters. 35 days, it should be 17 millimeters. So study that, just make sure you memorize it. I might have a pig laying out on a dissecting pan with a measurement tape beside it, and I want you to be able to look at the measurement, see what it is, and then determine the pig's age off of that. So do memorize that. Then on the last page, the back page, this is all I used. Basically everything we did in the rat, everything we did in the cat, I just want you to find it in the pig. The only difference with the pig though is that it says umbilical arteries and umbilical vein, and I believe that's it. So if you look at this, if this is too dark, there's other pictures on Google, or like I said, that video, this is what I could possibly ask you of the pig, but nothing more, other than obviously the size. And that's pretty much it that I want you to know from here. Also though, be able to identify if it's a male or a female on that pig too. So beside the size, that's easy. Same way as a cat. Does it say it in here actually? Let me see. I actually don't know where it says it. It's good. You don't have to figure out the gender. We're fine. So then also, okay, so like what's due? So the extra credits will be due. If you didn't already turn in post-lab for the reproductive system, turn that in. Also, the questions, the genetics questions packet, that is also due. Also, next week we are going to be, I think we're going to do evaluations, so we'll do it before the practical. I'm just going to need someone to have to run that to the biology office for me, or department office for me. And then we can take and you can just run in real quick and take the practical. So yeah. I think that's it as in terms of questions. I gave you guys a lot of information this time because we didn't really have 4 chapters, and she doesn't want me to include the genetics in with the 25 stations. She wants it to be extra. So it is a lot more information, and it is a lot more detailed, especially since we have the endocrine and we have all those hormones that we need to memorize and all that. So it's going to be a lot. Basically, instead of 4 chapters, you kind of really only have 3, part of 26, but like really there's nothing it's 2 pages. So it's going to be a big chunk. I would definitely say the station that has 8 is either going to be reproductive or endocrine, but those are going to have a lot. Urine, not so much. Urinary, not so much, but the other 2 are going to have a lot included, definitely because of the mitosis part of the endocrine system. But if anyone has any other questions, or you didn't understand something I said or went over, just email me, let me know. In terms of picking up your assignments, please email me, get with me sometime on Monday. I will be... I will be in Supple from 11 to 12. And I will be in the, where will I be? I will be in, yeah, let's just make it the biology lounge on the 3rd floor. So I'll be in the 3rd floor in biology lounge with all of your grades, your quizzes, everything. Please come to me, stop by, and get it. That way you can study it for the quiz. Other than that, good luck, you guys. Email me for any questions, and I'll see you Tuesday.